I would like to welcome Gwenaelle Géléoc. She's Associate Professor at Harvard Medical School and Co-Director of Holt Géléoc Lab at Boston Children's Hospital. Today we'll be talking about designing personalized gene therapies for people with Ushersyndroom. For that, I'd like to welcome you to the studio, Gwen.
Thank you for having me.
Yes, nice to meet you. My first question would be, people with Ushersyndroom, they share a clinical diagnosis. Why then is it also so important to know which specific mutation underlies this clinical diagnosis?
Yes, that's a very, very important question. So there are actually about 12 different genes that are associated with Usher syndrome. And Usher syndrome is classified under 3 different classes depending on the onset and the progression of the disease. So to know what gene is affected and what mutation is associated with the disease, help us understand the clinical progression, the clinical diagnosis for the patient, but it also helps us think about, developing therapies that are specific for these mutation and these genes.
Yeah, and when we look at those therapies, before you've experimented with replacing entire genes, then I would think the specific mutation would not be as relevant, right?
That's very true. So there are different gene therapies that are explored in our lab and in many other labs in the world. One of them is what you call gene replacement therapy, where you're basically providing a new, fresh copy of the gene. In this case, it's true. The mutation doesn't matter as much because you're providing a brand new gene. Now, the issue is that this treatment is only amenable to genes that are small enough that they can be packed into the viral vectors that we use in the lab. You can think about it as, for a small gene, it fits into a small car. If you have a big gene, you would need several cars. And that gets just a lot more complicated to provide that gene. So for larger genes, the gene replacement therapy is not an option. And so we have to look for other options. Some of them might be using the new tools that we have, to do gene editing, basically using, you can think about it as, scissors that are going to go into the gene and then cut the region where the mutation is and repair it. So that's one option. And the other option is the use of antisense oligonucleotide. We call them ASO, others call them AONs. And these are basically short oligonucleotide sequences that will target a specific region of the gene where the mutation is. And sort of like you can think about it, put a Band-Aid on that mutation.
Okay. And then you can target specific mutations for specific Usher patients.
That's right. So in this case, you're able to target a specific mutation in a specific gene. So to do that, it's important to have a really clear genetic diagnosis and know what the patient, what gene is affected for each patient and what is the mutation that leads to the disease.
Yeah, and I can also imagine Usher syndrome already is quite a rare disease, and they are focusing on specific mutations that maybe a handful of people have.
That is...
How are you able to validate your efficacy and safety, for example?
That's very important. So it's true. So Usher syndrome is considered a rare disease. And obviously, if you think about a specific mutation in a specific gene, you're not looking at a much smaller population. So in the US, we have a way of developing therapies for disease that are associated with a limited number of patients. We call them N of 1 or N of few clinical trials. What you need to for that is to perform the preclinical studies in lab. So you might use cell lines. We actually use patient-derived stem cells, which we can use to develop mini organs in a dish. So we call them organoids, inner organoids, or retinal organoids. And these patient-derived cell line can be used to test the therapies. So this is the work that you do in a lab. And then after that, the idea is potentially you could validate the function using an animal model. So some labs have used zebrafish or others have used mouse models. And once you have basically a portfolio that demonstrate the efficacy of your drug, in addition, perhaps in an animal model, then you can go to the FDA and request a N of 1 clinical trial, which means that you're going to actually provide the therapy to maybe one or a few patients and validate that this therapy is actually efficient for these patients with this mutation, and then make this therapy available to other patients who have a mutation in that region.
Yeah, and how is that different from other clinical trials? Why is it so challenging, for example, to do a randomized clinical trial that's quite common in other kinds of diseases?
So randomized clinical trials are good when you have a larger population, right? Because then in that case, you can provide either the drug or a placebo drug and then really compare the outcome and validate that your drug is making a difference versus a placebo drug. If you don't have a lot of patients, you don't have the possibility of doing that kind of randomized clinical trial, which is when this N of few or N of one approach is really important.
I think this research still needs to be continued, right? It requires a lot of collaboration from researchers. I think it's a good opportunity today at the USH 2025 conference to kind of connect people, but also researchers as well as the patient community. You've actually been part of the organizing committee. And why do you find it so important to actually connect the scientific as well as the patient community of Ushersyndroom?
It's really crucial for us to come together. I think this conference, and this is the third time that I am involved with this conference, that has the scientific days for a couple of days and then the patient day. But even though these are two events, they actually are together. So we're going to have a lot of patients present here for the science part, and most of the scientists will still be here on Saturday when we have the patient day. And I think it's very important for, first of all, for all the scientists and the clinicians to come together and discuss the progress they've made using different approaches. We can learn from each other. New collaborations will come out of this meeting. And it's also important for our trainees, our students, our postdocs who are coming and presenting posters, giving talks. It's good for them to be exposed to other scientists, but also, very importantly, to the patients. So they understand what their work is about. It's no longer just working on a cell line. They're actually seeing patients and understanding what it means to be deafblind, what it means to have perhaps a cochlear implant, what it means to use sign language, and perhaps tactile sign language. So one of the first conferences I organized was in Boston in 2014, sorry. And for this conference, that was my first time really being surrounded by patients who had Usher syndrome. And what really struck me is to see the difference between the kids who had Usher syndrome and had been provided cochlear implant, which allows them to develop, fairly good communication, and they can hear quite well. And comparing that with patients who were from the previous generation, before the cochlear implant were available. And these patients were using tactile sign language. And that was the first time I actually witnessed tactile sign language. And I was just amazed by the way these people were communicating. In particular, they were actually learning new words because we were talking about the science, and the translator were basically creating new words on the spot for these patients.
I can imagine this has brought a whole new perspective on your work, and it's cool also to see the development also in your work, literally in Usher patients. Now, the Ushersyndroom Foundation has set a moonshot goal of making diagnosis and treatment standard of care worldwide. Finally, could you tell me, how does your research contribute to making this goal a reality?
Yes, so the PUSH program is really exciting, and it really the idea is to go from the clinical diagnosis to the genetic diagnosis, determining which mutation, which gene is involved, doing the testing in a lab to develop therapies, and then bringing them to the patients. It takes a lot of money, it takes a lot of time, it takes a lot of work, and we're hoping to be able to streamline this process, perhaps driving industry partners to really allow this to go faster and being amenable to many more patients.
We'll still be in touch, Gwen. I'm looking forward.
Absolutely. Thank you.
Thank you so much.